Good evening. My name is Julie Mickleberry. I'm the Associate Director of UC Santa Barbara Alumni Affairs, and it's my pleasure to welcome you, our alumni and community members, to the opening lecture, The Promise of Longevity, Science and Technology, the first in our 6-part Aging and Longevity series. When Dr. Nicole Albata, thank you for letting me know. Is that better? Okay, wonderful. When Dr. Nicole Albata approached us with the idea of creating a community lecture series to showcase groundbreaking research on successful aging and living longer, healthier lives, I was immediately on board. We are fortunate to have world-class researchers tirelessly working to extend healthy lifespans, and it is an honor to partner with them to share this incredible knowledge with UCSB alumni and friends. I want to thank the talented faculty of the Center for Aging and Longevity Studies, and with the dedicated efforts of Diana Thomas and Dr. Nicole Albata, we were able to collaborate with Dr. Joel Rothman, UCSB Development, Direct Relief, and the Santa Barbara Independent to bring you all here to explore aging and longevity through the diverse lenses of biology, neuroscience, psychology, engineering, and anthropology. Many of you have already signed up for the entire series, which is fantastic. There's still room for many of our lectures, and I would encourage you to share the information with your friends after this evening. For those of you who have not, you can actually sign up right now if you'd like. There's a postcard outside of the check-in if you haven't had one, if you haven't grabbed one. I know that this series promises to leave us all more informed and perhaps inspired to embrace the aging process with optimism and curiosity. And so without further ado, it is my pleasure to invite Interim Dean of Division of Mathematical, Life, and Physical Sciences, Dr. Shelley Gable, to the podium to introduce our amazing speaker, Dr. Rothman.
Thank you. Hello, can everybody hear me? Okay, great. It's my pleasure to be here representing the UCSB administration and particularly the Division of Mathematical, Life, and Physical Sciences, expressing our enthusiastic support for this lecture series and for the Center for Aging and Longevity in general. It's one of our leading edge research centers at the university, and we're very proud of it. CALS brings together hundreds of scholars and researchers from 17 different academic departments across our campus, spanning a broad array of disciplines. And each of their faculty and researchers are dedicated to facing and addressing the challenge that's of vital importance to all people, which is to harness the promise of longer, healthier lives. This center positions UCSB at the forefront, and as obviously, I think also the most interdisciplinary institutions in California and the nation that is focused on advancing longevity science and technology. And it will take an interdisciplinary approach to address this challenge. So without further ado, I'd like to introduce our speaker for tonight. Dr. Joel Rothman is a distinguished professor, and he is the Wilcox Family Chair in Biotechnology. He's also the founding director of the Center for Aging and Longevity Studies at UCSB. Dr. Rothman received his doctoral training at UC San Francisco and the University of Oregon, and was a postdoctoral fellow at the Medical Research Council in Cambridge in the UK. He has served as the chair of the Department of Molecular, Cellular, and Developmental Biology, and also as the director of an interdisciplinary graduate program called the Biomolecular Science and Engineering Program, also at UCSB. He has also served as a professor at the University of Wisconsin-Madison and the University of Auckland, New Zealand. This part I didn't know about you, but prior to graduate studies, he was a winemaker at Buena Vista Winery in Sonoma, California, and a public radio classical music announcer. So, for over 3 decades, his research laboratory has investigated aging, longevity, and regenerative medicine, supported by research grants from the National Institute on Aging and many other agencies. He is also an elected fellow of the American Association for the Advancement of Science, which is no small feat. So please join me in welcoming Dr. Joel Rothman for an engaging lecture on aging.
Thank you so much, Dean Gable, who I knew as Shelley until a few months ago when she became my dean. Well, thank you all for coming tonight this evening. It's really a pleasure to see all of you here. And I hope to share a little bit about my excitement about the science and technology that is really going to change the arc of the human experience. And I hope I can convey that today. So before I get started, I want to express some thanks to some people. First of all, to Alumni Affairs, in particular, Julie Mickleberry and Samantha Putnam, who unfortunately couldn't be here tonight, but Julie has done an enormous amount to put this whole series together. We're so grateful to her, and she's here. You might express your thanks to her if you see her. And the Center for Aging and Longevity Studies, CALS, Nicole has done an enormous amount to get CALS off the ground in so many different ways and also help organize these series. She is also here. Thank you so much, Nicole. And I want to advertise her talk, which is coming up at the very next talk here. Nicole will be given a talk in this room on February 27th. I strongly urge you to go to it. It's going to be very interesting about psychology of aging. And also Diana Thomas, who is also here, who provides administrative support for the center, and we're really grateful for that. We also are deeply grateful to Direct Relief for providing the venue and the support and helping organize this program in so many ways, in particular Martha Vasquez, who's done so much to put this together for us. I really appreciate all of that, and we also are grateful to the Independent for its help with us. I need to thank my council of advisors, deeply grateful that these four people have come together to help advise us on sort of the business aspects of a center. I'm a geeky scientist. I don't know how to handle business stuff, so they have been enormously helpful to us. So what I'll say is three of them are alums, actually. Sue Wilcox got her PhD here. Mark Schwartz and Fred Steck also got degrees at UCSB. They are tremendously supportive of the center, and I rely on them deeply for their wisdom and their support. And I can't express enough thanks to all of them for that. And I will also say that two of them were also directors of chairs of the board for Direct Relief, and that's Angel Iskovich and Mark, who both ran the board for Direct Relief. So they have a strong connection here. And finally, I have to thank Sue one more time for providing my endowed chair, which I'm deeply honored to have, which has supported much of my research activities. So thank you to those four, all of you, for helping me so much with the center. Okay, so what is the Center of Aging and Longevity about? So I was trained as a biologist, a developmental biologist, thinking about how time affects biology, how a fertilized egg over time turns into a human. And the intersection of time and biology is also faced in a very different context. Turns out it's not so different, but and that is the interface of time and biology causes us to age. And of course, time progresses, biology progresses through time, and it causes changes, as we all well know. Let's see. I guess I have this here. So we know what happens over that time period. And so we are really interested, the center is interested in understanding the interface between time and life, biology. And I've been interested in this problem for a long time personally in my own research, but I realized that the science was burgeoning so dramatically. The advances are so startling, particularly in the last several years. The advances in this field are breathtaking, and I'll give you a little flavor for that. And that led me to realize that it was definitely time to start a research center around the problems of aging and longevity at UCSB. And I am delighted to say that center has taken off and is really burgeoning now. We cover 6 major different research areas. We are one of, as Shelley said, the most interdisciplinary centers for aging and longevity studies, I think, anywhere. We span from biomedicine to engineering to computer science to social sciences, psychology, even the humanities and fine arts. We have 37 faculty from 17 different academic departments, more than 350 affiliated researchers now studying all sorts of aspects of these problems. And so it's really an exciting time, and I'm deeply grateful to the university for supporting this new activity for us. This center has been going for only about 3 years or so. And what our driving goal in the center is to deal with aging and longevity in many different ways, but particularly let's focus on this element. And that is, as you all know, one of the things that is very striking with age is the onset of age-related diseases. In fact, a huge number of diseases have at their root aging. Cancer, heart attack, stroke, Alzheimer's, etc. We're healthy up to a point, and then those things kick on. And if we can understand what is happening to predispose us to those diseases, if we can get at that and we can slow down the rate or interfere with that in some way, we can treat all diseases simultaneously instead of treating one at a time. We can get at the root. And that's really what we're driven to do. We're really developing a new kind of preventative medicine against all age-related diseases. The goal of CALS, and I want to be clear about this because there's often confusion about this. The goal of CALS, often when you think about extending longevity, people think about something like this, and that's how we extend longevity to a large degree nowadays. The goal of CALS is not to do this. We do not want to extend the period in which people are suffering, in which they're on life support and so forth. We are not interested in that at all. The goal of CALS is to do this, to bring youth and vigor, continue to bring youth and vigor to people for as long as possible, to extend the period of health span, to extend the period in which they're healthy and vigorous. That's what our goal is all about. We may also, as a subsidiary effect, extend lifespan to some extent. That's a reality, but that's not our driving goal. And certainly not to extend the period of life that is unpleasant. The goal from all that is to make all people live better longer. Sue will appreciate that line. Okay, so we know right now about a bunch of things that we can do. And I'm going to very quickly review things that many of which you already know about. In your own life, you can extend your healthy lifespan through exercise, very potent, very powerful element, proper diet and weight. Caloric restriction is another one. So if you actually cut your calorie intake way down, I'm not talking about obesity now, I'm talking about way down, and this is true in every animal that's been looked at, you will slow the rate of aging, and in fact, it'll extend your lifespan. Some people say, well, if you're actually living right at the edge of starvation, it's not really true you live longer, it really just feels that's brilliant. But in fact, this was just published last year. The rate of aging goes down for people who are caloric restricted by about 20% only compared to people who are not, and those age more quickly. So that's another powerful intervention. Some of you may have heard about the blue zones. The blue zone lifestyle is and I won't go into the details, but you can look it up online, a powerful correlation, at least, for people who live longer. And sleep is certainly an important contributor to extending lifespan. So all of those collaborate. Those are all things you can do right now. And there are things you can avoid. For example, we all know smoking is not going to extend lifespan. It's going to do just the opposite. It has a big effect. At age 35, if you quit at 35, you'll immediately extend your life by 8 years on average, of course, across the population. Now, there's another one I hate to talk about, especially as a former winemaker. I was the winemaker at Buena Vista Winery. This breaks my heart because for all these years, it was okay, right? In fact, it was good to be a moderate drinker. But the evidence is becoming clear that not only things like cirrhosis of the liver and so forth are associated with alcohol, but in fact, alcohol actually increases the rate of aging. I hate to say it. It's true. I've cut my own wine drinking back. So now, instead of a routine, it becomes a special thing that I have on the weekends. This study, for example, showed that there is a direct measure in the rate of aging with people who are having 32 units of alcohol, 32 drinks per week versus those that have 10. It actually does change the rate at which you age. Bad news. You're going to hear more about this if you go to a talk on May 22nd in this very room. One of the members of CALS, Dr. Karen Shulimsky, will be talking about her studies of alcohol-induced neuropsychopathology that occurs in later life. So if you want to learn more about this, I would go to it. If you don't want to think about it because you want to keep enjoying your alcohol, then don't go to the talk. Okay, so Okay, so those are all things that we can, please don't do that, computer. There we go. That we can, sorry. Are you going to run? Yes. Okay. Those are all things that we can do now, but the science is going deeper than this, and that's what I'm going to talk about a lot. So our goal is to figure out ways to slow down the aging clock. Of course, the aging clock goes at a certain rate, and that's why we progress to looking not like the picture that was shown at me earlier, but looking like this guy, and that was taken a while ago. What we want to achieve, and in fact is achievable now, this is achievable in model animals, is to slow the aging clock down and extend the period of youthfulness, delaying the rate of aging. And that is doable. And I want to give you evidence for that. One way to do that is those behavioral things that I talked about. But also, and this is really why, if you Google anti-aging, you're going to get tons of stuff. So one possible approach is to use supplements. There is a problem with supplements, and there's a problem with Googling anti-aging. Most of what you get is garbage, frankly. Charlatanism, it's widespread out there. A lot of it is potentially harmful. This goes way back. So this is the emperor of China who built the Great Wall of China and the terracotta soldiers. You may have heard about this. And he was fixated on wanting to live a long time. So he had people consulting him on that, and they gave him anti-aging supplements, including mercury. So he didn't live too long. And just last year in Japan, this was a story that came out just June last year of a supplement that killed at least 80 people. It was supposed to be an anti-aging kind of supplement. So we have to be really careful for this and about this. And as a result, one of the things we're doing in CALS is creating a clearinghouse of reliable information that the public can go to and look for scientifically validated, rigorous studies that back up whether a certain intervention is appropriate or not. That effort is led by Dr. Pradeep Joshi, CAL's Director of Scientific Communication, and it was made possible by a generous gift from Mark and Linda Schwartz. I don't have a picture of Linda here. Mark is sitting right here in the front row, and we deeply appreciate getting this program off the ground. We will have a public release of this later this year, and that website that provides all that information will be available to the public at no cost. This is a public service. Okay, so talking about supplements, and we'll be talking about that a lot in the clearinghouse. People think about supplements all the time in anti-aging, and Consumer Reports issue just last September had a story about it, 7 anti-aging supplements to try. The thing that jumped out at me, for me in that study, or in that article rather, is that the 7 they recommend, these things right here, this thing is falling off my ear. These things right here are nothing new. There's nothing new here. We've known about this stuff for ages. So, it's a useful article, but there wasn't anything new. That's changing. The other thing to say, by the way, is none of these slow aging per se. They don't slow the aging clock. But that may be changing. There is a supplement that was reported very recently, just in 2023. It's been around for a long time. But studies showed that this compound called taurine, which is something you make a lot of, you have a lot in your body, and you eat tons of it in your food. Taurine does a funny thing as we age. The level of taurine in all creatures, including humans that were looked at, the level of taurine drops with time. Actually, that's not shown. It drops with time as we get older. And so these the people who studied the effects of taurine then asked, what happens if you supplement with taurine? And when you do that in little worms or mice, you extend their lifespan. So this is a lifespan curve. This is the number of mice that are alive at a given time. And that dotted line is without taurine, and this line is with taurine. It extended their lifespan. It did not only extend their lifespan in both creatures, it slows the rate of aging. The signs of youth last longer. They have lower rates of disease, age-related diseases. So all signs of aging are improved taking taurine for these animals. Now, this hasn't been validated in humans yet. I will confess I take it every morning. I take 1,000 milligrams every morning. I think the risk is extremely low because we get tons of it from our diet. If you drink Red Bull, you're getting a bunch because the bull is taurine. That's where Red Bull, the name comes from. It may work. We don't know how much. Okay, so let me just summarize what I've said so far. And please don't take these at face value. These are rough numbers, and the bottom ones are fantasy. But these various characters, these various things I talked about can extend lifespan. And for sure, these are in rigorous scientific studies by substantial amounts, up to 14 years in the case of exercise studies, for example, or a dozen years or so forth. And these other things are not as well known. There is good evidence for drinking, and caloric restriction is that why there's a question mark there. Taurine, not known yet, but I threw out 3 years randomly, but we don't know what it's really going to be. But we can slow down the rate of aging with these things for sure. Now, having said that, I want to put all that aside and talk about the science and technology, because the science is really blowing open. And this is why I decided it was time to start Center for Aging and Longevity Studies. Really shocking, startling discoveries that are coming out, especially in the last few years. This science promises to get way out ahead of what I just told you about. And let me justify that. Let me tell you why I'm saying that. So first of all, the field is enormous now. This is on the cover of one of the most famous and prestigious scientific magazines called Science, that had an article on aging several years ago. But there's a huge number of papers that have come out since that. In fact, right now, as of today, 700,000 papers on aging and longevity. I'm trying to catch up and read all those. A little hard to do. The problem is it's about 1,000 a week now. So I have to update this thing constantly. 1,000 papers a week. And this is going to reach 1 million in less than 5 years. Studies about aging and longevity that taught us some aspect of what happens and how we can intervene in it. Okay, so let me get at the root. What I think is sort of the most global contributor to aging and longevity. These things that I've talked about are tweaks. The big thing, ultimately, is our genomes, our genetics, our chromosomes. That's the big thing. Genes determine longevity. We knew this before ever doing any science. Because if we look at species, we see them age at very different rates. This wonderful little creature and that little wonderful creature have very different lifespans, 12 to 15 years versus 80 years or more. Why? The whole difference between those two is their genomes. This has a dog genome, tells it how to become a dog. This has a human genome. The genome that controls your identity as a species also controls your rate of aging. And so we can take advantage of looking at the natural world and make some predictions, or at least some guesses. This is a range of animals showing different lifespans. I've sort of tabulated some of these. The shortest living animal that we know of is the dainty mayfly. Females become adults and live 4 hours. That's it, 4 hours. Can you imagine? I'll talk a little bit about these little worms. They live about 3 weeks, which is convenient. As you go up, you see humans are kind of in the middle here. Life expectancy around 80 years. We can go higher. And so one of the things to see here is that we can go higher. Here is a person who did go higher than 80 years. Some of you may have heard of Jean Calment, who lived until 1997, 122 years old. At age 40, she looked pretty good. Looked pretty good at 80, I thought. 121, well, I mean, for 121, she looked pretty good. Now, Jean Calment, that's pretty remarkable. She lived to 121. She was a smoker, and she drank. So that goes against everything I just said, right? Here she is at 117. Now, having said that, she did quit smoking at 117. So, that's why she lived to 121. Okay. Now, thinking about the human population and this variation, of course, a lot of people don't live anywhere near 122 years old, even though they did everything right, and she didn't. It's very important to think about the genetic variation and the variation in environment and so forth on humans, the variation across humans. And I want to advertise one of our members of CALS who's sitting back there, Mike Gerben in anthropology, who is going to be presenting on these people. That's April 24th, right in here. This group of people called the Chimane people who have very low rates of a number of diseases. They seem to have extraordinary health over aging. And Mike has done just fantastic, very exciting work to figure out what the basis of that is. And if you want to hear more about that, please come to his talk then. I think it's absolutely fascinating. And so lots of different diseases, slower rates. I'm not going to give his talk for him, so he can do that. Okay, so that's humans. Humans are kind of in the middle. Now, they are in the middle because there are creatures that live substantially longer, 150 years for a tortoise. A bowhead whale is a mammal like us. Bowhead whales can live 200 years, so I call them Jefferson, because there are bowhead whales on the planet today that were swimming around when Thomas Jefferson was president, right? I mean, that's a pretty weird thought, but it gets weirder. They're mammals. There are vertebrates, that is, they have a backbone, a skeleton like us. The Greenland shark that can live half a millennium. Think about that. And so I call these guys Magellan. You can figure out why. There are Greenland sharks today, probably, who Magellan could have sailed over, the same shark 500 years ago. Think about that. And that shark actually has been in the popular news in New York Times that was mentioned last September, because its genome, the code of its genes has been decoded, and we can get a lot of information from that. We can look at what is in its genes that allows it to live 500 years and maybe learn something that we can apply to humans and make humans live longer. Maybe not 500 years, but certainly do something about that. It's very interesting. And if you have questions about what we already have learned about that, I'd love to talk about it. Sorry? Okay. Okay. So genes have something to do with it, but what's the risk? This is all correlative. What can we actually show a causal effect? The answer is yes. Genes do extend longevity, and the experiments that revealed that were done initially. Some of the most important experiments were done in this little creature, this simple little creature, a little worm called C. elegans. The movies don't matter. This is a pretty famous little worm to experimentalists. There it is. It's one of my favorite creatures after humans and dogs. I've spent my whole scientific career pretty much working on these little guys. You may not have heard of these, but they've been very famous and contributed a huge number of advances in biomedical science, working with these simple little animals you can grow in the laboratory. In fact, they have people working on C. elegans just in the last 15, 18 years or so have won Nobel Prizes. 8 Nobel Prizes now already awarded to investigators working in this little creature, including last October, 2 of my colleagues received 2 other Nobel Prizes. So, and that's because of the importance of these organisms. Now, these are perhaps the most important organism for understanding what causes aging and longevity. And that started with a discovery of a single gene change that did a remarkable thing. This was Professor Cynthia Kenyon at the University of California, San Francisco, discovered that a single gene change could extend the lifespan of these animals dramatically. In fact, if you do the right experiments, you can extend their lifespan by a factor of 10. That would be like a human living to 700, 800 years with one gene change. You make a single change, one letter of the 100 million letters in all the genes of this animal. One makes it live that long. That's quite remarkable. Now, that's a worm, so who cares? I'll tell you who would care is humans, because in fact, centenarians, people who live to 100 or longer, have a high frequency of changes in their genome when you look at their genomes in the same genes that are affected, in the same gene that's affected in this animal. So it's not just a worm. Humans also respond to that gene change in the same way. Probably it's correlative, but it's probably true. Okay, that's gene changes. You can't just go in and change your genes very easily. You can change your pants, but can't change your genes. Oh, that's terrible. Sorry. So my daughter wasn't very grown, so that's what I did. But if you get a discovery of a gene change like this can point immediately to drugs. If you know a certain gene change causes an effect, you can start to design drugs that have the same effect as the gene change. And a lot of drugs do exactly that. And in fact, these kinds of discoveries in experimental animals have led to the discovery of drugs that do the same thing as gene changes that extend health span and lifespan. And I should have said with those long-lived animals, they stay young 10 times longer. It's not just they're not just hanging on life support. They stay young 10 times longer. We slowed the aging clock. Okay, here is one of the drugs that came out of these kinds of studies. Some of you may have heard of it. It's called rapamycin. It was discovered originally in Easter Island. It's called Rapa Nui, hence its name. It's been used in the medical profession for many years as an immunosuppressant to prevent tissue rejection. But in fact, it was found, because of these kinds of studies that I mentioned in the laboratory, it was found that it also extends lifespan of every animal that's been looked at. Across all these different animals, across all of evolution that's been looked at, this range of evolution, it extends their lifespan and their health spans, keeps them young longer. It's currently in clinical trials in humans. We will know how effective it is quite shortly. Some people are taking rapamycin. I would if I were a little older, and I may start taking it in a few years if the clinical studies really bear out what's happening in every single other creature being looked at. Currently, rapamycin is not the only one. There are over 100 clinical trials going on right now for interventions that slow down the rate of aging. Over 100 clinical trials. So stuff's coming out like crazy that's going to come onto humans if any of these things succeed, I think, in the relative near term. Certainly not in 50 or 100 years. We're contributing in cows to that quite substantially. We have one of the most advanced high throughput facilities. This is what it looked like just next to my office in the Bio2 building for robotically driven experiments that can look, instead of looking for a single drug at a time or a single experiment at a time, can do thousands or tens of thousands of them overnight while a graduate student sleeps. So we have this tremendous facility for doing enormous numbers of experiments simultaneously at UCSB. We're in the perhaps top 2, 3, or 4 institutions in the country that have this level of high throughput facility. I'm proud to say this just started this last year with a large grant that the university was able to get. And that's going to turn up a lot of really cool science. Okay, so that's drugs, and this will allow us to do drug discovery for sure, including drugs, by the way, that may not be profitable because they're repurposing, like rapamycin is repurposing known drugs. But another thing that has been a huge advance in the last few years is that we now can measure how fast we're aging and thereby look for interventions that might change the rate of aging in a relatively short term. So if you had to look for lifespan extension, if you want to do that in a worm that lives 3 weeks, no problem. But in a human, you don't want to wait 80, 90, 100 years to see if it works. And we now have very powerful ways to measure the rate at which we age. And that is because our DNA, our chromosomes, receive little marks on them. They receive little chemical bits that are added onto our chromosomes. And those chemical bits change as we age. And you can use statistical and computer methods to determine the rate at which someone is aging based on statistics, correlation with people at different ages, different biological ages. So it gives you a very accurate measure of your effective biological age, not your chronological age, but your biological age, effectively how old you are biologically. And this has really revolutionized this science as well. So with this kind of aging clock, we can see that certain behaviors slow down the rate of aging, healthy food intake and exercise. You can actually see that on these marks in your chromosomes. Huge statistical power because there are hundreds of thousands of these little marks that are added on the chromosomes. And the things that are bad, stress and infections and other things, overweight and so forth, speed up the rate of aging. And you can see that directly in this aging clock. Now, this is already becoming accessible to the general public. You can send off a sample of cheek or some blood, and you can measure your rate of aging. If you want to see a certain intervention slow down your rate of aging, you can do that yourself. Soon we'll have phone apps that will do this. You're age 52 years and 5 months. We're not there yet, but that's coming. My Apple Watch, for example, will have that kind of feedback available coming. And 2 of the investigators in cows are working on these chromosome modifications, these chromosome marks that are associated with the aging clock. And I want to mention both of them. They are not talking at this series this year, but we hope they will in future years. All right. So now we have a clock that we can measure. This is very important in clinical trials. It's very important in our own behavior because we can ask which things slow down the rate of aging. But now I want to get into a really crazy area that I wouldn't have imagined was possible until a few years ago. This is something that has really advanced just shockingly in the last several years, and that is the following. So we know that most of our cells age with a clock that's spinning around at a certain rate, and the clock goes forward, right? But we humans have two different types of cells. We have all the cells we know of that are feeling like they're getting old and tired. But we have a different type of cells that do a weird thing. We have a type of cells that do that age backward. They de-age. What? Come on. How can you de-age? But this has to be true because every time, regardless of the age of the parents, a new baby is created, an embryo. The thing isn't old. It started fresh again. The cells that came from the older parent and made the new baby got refreshed. They got youthified. We're in my lab right now studying that process. We're using computational techniques to understand that. We and, am I losing power there? We and certainly other investigators, and we're following on this, have found that embryos themselves get younger as they get older. The first early phase of embryos actually start de-aging, and then they start aging forward again. Now, this means that if we really understand the difference between these two kinds of cells, we might be able to achieve something quite remarkable, which is to start aging backwards. Some of you may have read this short story by F. Scott Fitzgerald or seen the movie, Benjamin Button, and that 70-year-old man who ages backwards until he's a baby. In fact, Benjamin Button is not science fiction, not now. It is for humans currently, but it might not be always. And that is because we can intervene in aging in ways that actually make organisms age backwards. And part of the way that's done is through gene therapy, going in and changing the genes in the organism, ultimately would be us, changing the genes in a way that triggers something to happen to change the rate of aging or even get it to back up. I want to show you how this actually, one example of what this looks like. So I work in my lab. We also work on mitochondria. A couple labs work on these mitochondria, which you may remember from high school biology. These are these little things inside cells that are the powerhouse of the cell that provide our energy. They have their own DNA. So most of the DNA is inside the nucleus. That's where we think of as the human DNA being. But these guys also have a little DNA, which is a remnant of the fact that mitochondria were once bacteria that went inside a primordial cell. And we are hybrid species. We came from two different evolutionary origins. We're not a single origin of evolution. We're two, because we came from mitochondria and from the cell around us. Their DNA breaks down over time as we age. You can mimic that in the lab. Mice with normal mitochondria look like normal mice. And the investigators who worked on this did a little trick to make the mitochondria defective. Okay? And so they have a little trick to make the mitochondrial DNA, the DNA inside there, bad. And what happened? They get mice that look like that pretty quickly. A few months, they look old, wrinkly, etc. Now, look what happens when you bring back the normal mitochondria. They restore the normal mitochondria. Those mice look like that. That looks like reversal of aging. It's not really as extreme as it looks, but it sure looks like the reversal of aging. And with just that study, it might not have been enough. But now, before I say the further steps, I want to say that my lab is working on how to get rid of the bad mitochondrial DNA, bring in the good ones. Chris Richardson, who's also in the Center for Aging and Longevity Studies, is doing the same thing for the DNA that's inside the nucleus, the human genome, because that also breaks down. And Chris is contributing to that in the center. But now, in the last few years, gene therapy studies on mice, that is changing genes in mice, have revealed a very remarkable thing. And that is that you can get mice to age backwards with the expression of certain sets of genes. 4 genes, you express them over and over, and you can take an older mouse and make it look younger if you just express those genes. There they are. That mouse is one that didn't get those genes. That is the same age it did. It did. And all of the features you look at for aging are reversed in those animals. You can actually reverse signs of aging with this gene therapy. you're actually backing up aging. And we have been doing these studies in my lab as well. With these little worms, we found that we can do the same thing. And the worms we can move much quicker than with a mouse. So we're really excited about that. And that project being driven by one of my graduate students was supported by Fred Steck, one of the members of the advisory board for CALS. So we're grateful to him for providing that fellowship. Okay, another thing that happens as we age, something else that we can tackle, is that we accumulate cells that are called senescent cells. These are cells that are not particularly useful or productive. As we age, they accumulate. So older people, it's supposed to be indicating, have higher numbers of these senescent cells. Younger people have very low levels. These are cells that just sit there as zombies and send out stuff that make us age. They don't do any good for us. They just make us age. If we get rid of those guys, in fact, we could slow the rate of aging, and in fact, that works. In mice, in this study, in mice, the investigators figured out a way to destroy the senescent cells. These two mice are the same age. This guy has the senescent cells. This one, the senescent cells were killed off. They live longer. They are healthier. The onset of age-related disease is delayed in these mice. So if we can get rid of senescent cells, then actually that's another way to tackle the problem of aging. And that, in fact, is a problem that's being studied through a number of investigators in CALS. In chemistry, Armin Zakarian and his collaborator, who's also in CALS, Cesar Cardenas, are developing compounds, drugs, that kill off senescent cells, eliminating the senescent cells in a mouse that's aged, trying to make them younger again. And they have some promising leads already. There's another way to get rid of those senescent cells. Now, this sounds weird, maybe, but that is vaccination. You can actually train your immune system to go after any kind of cell you want. So it is possible to vaccinate against senescent cells. Your immune system will attack them and eliminate them. When that is done in a mouse, same thing. Guess what? The mice age more slowly. It works. So we will, perhaps in the future, be vaccinated against aging. And that's not a joke. We can do it now in a mouse. Okay, one last major therapy that we can use is to take advantage of cells. We're made of cells, and we can get rid of cells by training other cells to get rid of the bad cells. In getting rid of senescent cells, we can teach our own cells to get rid of those cells. So here is a movie of actually a cell gobbling up that green cell. That green thing is gobbling up these other purple cells that are cancer cells. You can actually get them to gobble up and get rid of the cancer. This is a major area in cancer therapy now. It's being used in humans actually to cure a number of kinds of cancers. And but we can do the same thing by getting rid of those senescent cells. One of the CALS investigators, who many of you may know of, a very prominent researcher, Denise Montel, that movie is from her, who will be presenting here March 27th, is working on getting this kind of approach to work better with cancer cells, and that will also be applied with senescent cells. And could I pick up your question at the end? Is that okay? Yeah. Because I'm on a tight schedule. I'm almost done. Okay, so the last really breakthrough that's coming at us, and which CALS has in it going on, is artificial intelligence. Artificial intelligence, tremendously important in this domain because aging is an enormously complex problem, and computers help us enormously with it. And I want to just report on something that came out just this week. Some of you may recognize Sam Altman there, the founder of OpenAI, ChatGPT, and et cetera. His company is applying AI to figuring out how to make proteins do something better. So I said that there are genes that can make mice age backwards. So it turns out they've looked at those genes and they've figured out ways using AI to make those genes work better, just predicted based on the computer. And it looks like they're getting a 50-fold increase in the efficacy, driven by the large language models that are used, that are dependent on AI. So that is going to dramatically advance the science. And I will say that one of our investigators in CALS, a superstar young Professor Nina Mulane is working on the applications of AI to aging, and she's giving a talk here on June 26th in this room as well. So I'd encourage you to see that. Okay, I think I have another moment, so I want to tell you about where this might all go. So first of all, let me just say, before I go there, that all of this science, I mean, I wouldn't have believed this stuff 20 years ago or even 10 years ago, a lot of this. We're in a mind-blowing time. It's going to change humanity, I think, very significantly. And I often say, and I'm going to stick my neck out here, I think that the first person to live to 150 is walking around on the planet now. Possibly the students at UCSB, I've told them that it is quite possible that they're going to live to 150, and there's all sorts of societal problems. We can talk about it in the Q&A. But I think there's a good chance that'll happen based on the rate of progress of this science and science in general. But if their lifespans are extended that much, then we get into a very interesting place. Because imagine, this is the normal lifespan right here, all right? And imagine a 20-year-old makes it to 120. If they live as long as Jean Calment because of this science, what's going to happen in 100 years of science? If I look back in the past 100 years, it is mind-blowing. We didn't know what a gene was. We didn't know anything about cells. We didn't have any technology. The technology we have and the amount of information we have about how we operate and how we work as humans is breathtaking. 100 years, we know almost nothing. It's all happened in the last 100. So now, years. Now wind ahead another 100 years. With the technology we have only today, not the future technology that's coming, I just can't escape thinking that this thing is going to be pushed forward quite a bit. So a new technology will come. Some of the things I've talked about are perhaps already there. So that's going to extend their lifespans further. But now the lifespan goes further. Another technology can come along in that further extension of life. And you might see where I'm going. You extend life further, there is time for another technology to be developed and another. And ultimately, what happens if you are really able to extend lifespan along? The longer you extend it, the more discoveries that will be made, the more it can push it further. That is kind of a very exciting, but also very frightening prospect. And I think we all have to think about the impact of that. I think that's a very real possibility based on what we've seen with mice and with other organisms. Okay. So I'm going to basically summarize by saying, here's what's coming. We can track aging. We have longevity drugs. We have vaccines being developed against aging, cellular therapies, gene therapies, and artificial intelligence. All of these are going to be adding decades of healthy life, I think, to people who are even alive today, possibly. And the goal, of course, of all that is for all of us to live better longer. That's where we're headed. So the main message I hope I leave you with is that we don't have to face that expectation that the arc of life is what we thought it would be all our lives. In fact, UCSB and CALS are major participants in changing that expectation. So thanks very much for your attention. I'd love to take questions.